Hello and welcome to Chapter 22, Prokaryotes. That's bacteria and the Archaea. So in Biology 101 last semester, you all were introduced to the prokaryotes. Recall that the prokaryotes are single-celled organisms. They have a single circular chromosome that is not enclosed by a nuclear envelope. Prokaryotes include two of the three domains of living organisms. That is the bacteria and the archaea. The third domain is eukarya, of course. While the physical structure of prokaryotes is simple and while their lifestyle is, well, seemingly less than exciting, the effects of prokaryotes, effects they have on the biosphere, are very profound and long-lasting, as we'll see. Prokaryotes were the first inhabitants on the Earth. They appeared about 3.5 to 3.8 billion years ago. Now I say that prokaryotes are ubiquitous. That means they're everywhere all the time. They inhabit every environment from the moderate to the extreme. So you can find prokaryotes in the boiling hot springs in Yellowstone Park to permanently frozen environments like Antarctica, in salty environments like the Dead Sea, or radioactive regions like Chernobyl. Prokaryotes reside in the human digestive system and on the skin, and they're responsible for certain illnesses, but they serve an important role in the preparation of many foods, and we'll see that in this chapter, which is all about the prokaryotes. So our objectives here are to describe the evolutionary history of prokaryotes, discuss the distinguishing features of extremophiles, describe the basic structure of a typical prokaryote, describe important differences in structure between archaea and bacteria, identify the macronutrients needed by prokaryotes, describe the roles of prokaryotes in the carbon and nitrogen cycles, identify bacterial diseases that caused historically important epidemics, explain how the overuse of antibiotics may be creating superbugs, and describe the beneficial effects of bacteria to human health. So again, prokaryotes are ubiquitous, everywhere all the time. Prokaryotes inhabit all other living organisms, and so prokaryotic cells in your body outnumber the cells of your body. Your body's made up of about 30 trillion cells, and there are about 38 trillion bacterial cells inside of you right now. So prokaryotes are important in recycling nutrients like carbon and nitrogen, and prokaryotes were around long before eukaryotes. Prokaryotes, in fact, were the predecessors of the eukarya. Ancestral prokaryotes were obligate anaerobes before evolving into phototrophs. Now, obligate anaerobes, aerobic organisms, or aerobe means with oxygen. So an aerobic organism would be an organism like you and I that must have oxygen in order to survive. Anytime you put an A or an AN on the front of a word in biology, that means without. So anaerobe would be without oxygen. So prokaryotes, or ancestral prokaryotes, I should say, are obligate anaerobes. They lived in environments where there was no oxygen. Obviously, the early Earth, billions and billions and billions of years ago, was devoid of oxygen. And so the only thing that could live there would be an organism that didn't need oxygen to survive. That's an obligate anaerobe. Let's go to the first video, 3 Domains of Life by Mumu Math and Science. Really short video, simple little animation. I think you'll find it fun and interesting, and it's a quick look at the 3 domains. Now, prokaryotes are extremely diverse because they're extremely adaptable, and like I said earlier, they live in all environments on the planet. Now, we have a group of prokaryotes that we refer to as extremophiles. Now, phile means to love or lover of, You learned that in 101. We talked about hydrophilic, lover of water. So extremophiles are lovers of extremes. So they're able to survive and thrive, not just survive, but thrive in conditions that all other organisms, including humans, could never survive. So here is a nice little illustration, just giving you an idea, and I just want you to have an idea here of where you might find prokaryotes, and especially extremophiles. Here are deep sea hydrothermal vents. Now these hydrothermal vents spew hot water that's been heated by the mantle up, but they're very deep in the ocean. You have high pressure, high temperature. You have bacteria that are living there. Here are deep sea anoxic lakes. Anoxic, that's without oxygen. So here are areas that are devoid of oxygen. You have extremophiles. You have prokaryotes living there. Your polar regions, Antarctica, permanently frozen. Guess what? You have bacteria living there. Hot springs, these are sulfur-rich environments. Sulfur-rich, no fish can live there, but you have bacteria that live there. Soda lakes, hypersaline lakes, like out west here in the western United States. Guess what? Very little can live there, but you always have bacteria there. And desert environments, extremely hot, extremely dry, you have bacteria living there. And here is even nuclear contaminated sites like Chernobyl. Guess what? We think of radiation as being able to kill everything. You still have bacteria living there. All right? And so here's a little chart. I've adapted this from your text, showing you the extremophile group, just generic group, and the conditions for their growth. We have acidophiles, so these are organisms that like acidic environments, pH 3 or below. pH 3 is extremely acidic, like stomach acid, and below. Alkalophiles, or alkalophiles, rather, these organisms that like the other extreme of the pH scale. So pH 9, very alkaline or above. Thermophiles, now thermo has to do with temperature. So thermophiles like temperatures between 60 and 80 degrees C. So that's greater than body temperature. There you go, 140, 176 Fahrenheit. Hyperthermophiles, so thermophiles like temperatures above normal body temperature, which is 37 degrees C. Then hyper is above and beyond. Hyperthermophiles are going to like temperatures even warmer than that. So we're looking at above 80. Psychrophiles, on the other side of the coin, like really cool temperatures. So negative 15 to 10 degrees Celsius, right? That's well below freezing Fahrenheit. Most bacteria like it body temperature around 37 degrees, okay? But you have these psychrophiles that prefer it really cold. Obviously, these organisms are going to be living in like Antarctica. Halophiles. Halo has to do with salt concentration. So these organisms prefer high salt environments, like out in the salt lakes in the U.S. Southwest. Salt concentration of at least 0.2 molar. And osmophiles, on the other side of that coin, think of osmosis, the movement of sugar. So osmophiles like high sugar concentration. So you should have kind of some idea, and you can tell from the names here what they really like. Now this chapter is about prokaryotes, which includes bacteria and archaea, but often I'll use the word prokaryote and bacteria interchangeably. So here is a little illustration. You've seen a similar illustration like this before in 101. This is just a generic bacterium, and it just shows you all of the different types of characteristics that a bacterium could have. Not all of these things can be found on all bacteria, but there are some common things. For instance, the single circular chromosome made of DNA, that is common to all bacteria. Cytoplasm, of course, is common to all cells. Chromosome is made of DNA. There is a plasma membrane that's a phospholipid bilayer, just like the one that your cells have. There's a cell wall, which we'll talk more in detail about, and the cell wall varies depending on the bacterium. But the cell wall is rigid, and it gives that bacterium a shape and structure. It makes it more like a brick than animal cells, which are kind of amorphous. And then some bacteria have a capsule, not all, but some have a capsule. And then some bacteria have flagella. Here's a flagellum that spins around like a propeller and allows this bacterium to move through the milieu. And then these are pili. These little sort of hair-like structures in green that you see here. And they're just think of like Velcro. So they allow the organism to stick to surfaces. Okay. So all bacterial cells have a plasma membrane. It's a phospholipid bilayer. It's selectively permeable, just like the membrane around your cells. And all bacteria have a cell wall. Now that cell wall varies a little bit, and we'll look at the structure to differentiate various bacterial cells based on cell wall in a minute, but the cell wall is a protective outer layer that surrounds and protects the cell. It provides shape and rigidity, so it makes that little cell more firm, like a little brick, if you will. And it prevents osmotic lysing. So the osmotic pressure inside of the cell is extremely high. And if there was nothing to sort of surround it and hold it in, then that cell would fill up with fluid, and just like an overfilled water balloon, eventually it would burst. So the cell wall, kind of like the cell wall in a plant, prevents the plasma membrane from bursting, from lysing. We call that osmotic lysing, bursting at a high internal pressure. Okay, so cell walls are made of peptidoglycan. It's a protein and polysaccharide molecule, the main component of the cell wall, and we'll see the illustration from your text here in a minute. Now, there are different ways to distinguish between bacteria or prokaryotes. And so we have this binary selection platform that we use. And one of the components of that binary system is gram stain. Now, gram stain is just a staining technique invented by, of course, Dr. Graham. That really was his name. We call it the Gram stain, and it's a staining technique used to differentiate bacteria according to their cell wall composition. And so we use it's a stain counter stain system. So we stain the organism, the sample, twice, and depending on what color comes out, there it tells you if it's gram positive, gram negative, and that tells you what kind of cell wall it has. Okay, long story short. So gram positive, I usually denote that rather than writing out positive all the time, I denote that with the little plus in parentheses. So gram-positive bacteria have a thick, it's important for you to remember, a thick peptidoglycan layer. That thick layer is bound with lipoteichoic acid molecules, or teichoic acid molecules. They're like little ropes that hold the cell wall, the peptidoglycan wall, to the cell membrane. And the cell wall in gram-positive bacteria appears blue under the microscope. I'll explain that in a minute. And then on the other side, you have gram-negative. It's my little symbol for negative. Gram-negative bacteria, they have a thin peptidoglycan cell wall. They also have an outer lipid bilayer. So they have an outer bilayer and an inner bilayer. This outer bilayer contains porins. You're familiar with porins. They're pores. They're just little openings. They're little channels that allow things to flow in and out passively by diffusion, simple diffusion. Just like you'll recall the pores in the nuclear membrane. So they're just pores, porins we call them. And in gram-negative bacteria, the cell wall appears pink under the microscope. So let me explain that. So here's the illustration from your text. Gram-positive over here, gram-negative over there. So cytoplasm, plasma membrane, phospholipid bilayer, selectively permeable. Same thing over here in gram-negative. These little blue rectangles, this represents layers of peptidoglycan. You can see how thick this is compared to a single layer over here. And here's your teichoic or lipoteichoic acid molecules, which are like these little ropes, guy wires that are binding, linking, connecting the peptidoglycan cell wall to the plasma membrane. Okay? Thick peptidoglycan cell wall. That's what you want to remember about gram-positive. On the other side, we have gram-negative, cytoplasm, plasma membrane. There's your peptidoglycan there. You can see how thin it is. And so here is another lipid bilayer. And because there are two, we call this one the outer membrane and that one the inner membrane. And there's your porin labeled right there. So there's a little porin, a little channel that allows things to diffuse back and forth, in and out. And in the gram-negative, you have this lipopolysaccharide, this fat layer, lipid layer, on the very outside of the cell. So what happens in the gram stain? It's called crystal violet. You douse the slide with crystal violet. And you let it sit there for a couple of minutes and you rinse it off. Now, everything stains blue, okay? Everything stains blue. And that's what this kind of blue color is showing. So you rinse it off, it stains blue or violet, kind of in that range, that color range. And then you rinse that stain off and then you counter stain. So you use another stain called safranin, and you douse your sample. You're looking at a sample in a microscope slide, by the way. So you douse your sample with safranin, and the cells that have this lipid layer take up the safranin, and because you have just a very thin peptidoglycan layer, it gets blocked out. You don't see the blue peptidoglycan layer for the outer pink layer because it's so thin. So the pink color overshadows the blue color that you see over here. And so the point here is that gram-positive, because of their thick peptidoglycan layers, appear blue under the microscope, or stain blue, and gram-negative bacteria, because of that lipopolysaccharide layer and a very thin peptidoglycan layer, stain pink. And they look like this. Here's your gram-positive, and there's your gram-negative in pink. And here's an example, a microscopic sample. These are rod-shaped bacteria. We call them bacilli. One is a bacillus. And some of them stain blue or violet, and some of them stain red or pink. So we have both gram-positive and gram-negative in the same slide, and that's a very common occurrence to see both gram-positive and gram-negative bacteria in the same specimen. Right, but that's what they look like under the microscope. All right, another video, gram-positive versus gram-negative bacteria. This is from Neural Academy. Again, not a long one, but one worth watching. So if you watch the video, then you should be able to answer this very easily. Which of the following statements is true? And follow the instructions, check all that apply. And we'll look at the choices. So gram-positive bacteria have a cell wall anchored to the cell membrane by lipoteichoic acid. Porins allow entry of substances into both gram-negative and gram-positive bacteria. Cell wall of gram-negative bacteria is thick while the cell wall of gram-positive is thin. And gram-negative bacteria have a cell wall made of peptidoglycan, while gram-positive have a cell wall made of lipoteichoic acid. Okay, so let's look at these individually, and we're going to check all that apply. So gram-positive bacteria have a cell wall that is anchored to the cell membrane by lipoteichoic acid, right? And we know that that's true. Porins allow entry of substances into both gram-positive and gram-negative bacteria. Well, we know that only gram-negative bacteria have porins, so that's obviously not true. The cell wall of gram-negative bacteria is thick, while the cell wall of gram-positive bacteria is thin, and that is actually quite the reverse, right? The gram-positive have the thick wall, the thick peptidoglycan layer, and the gram-negative had the thin peptidoglycan layer. And the gram-negative bacteria have a cell wall made of peptidoglycan, that is true, while gram-positive bacteria have a cell wall made of lipoteichoic acid. No, all bacterial cell walls are made of peptidoglycan, so obviously that's not true. The only one true here is A. So with regard to reproduction in prokaryotes and in bacteria, you know this already from 101 when we talked about reproduction. Prokaryotes undergo asexual reproduction. In other words, there's no male and no female bacteria. It's just a bacterium. And it replicates its DNA and then it divides, right? We call that asexual reproduction. And the process here is binary fission, where the parent cell divides into two identical daughter cells, which then happen to be clones. And in case you forgot, here it is. You've seen an illustration much like this. Here's the parental cell. The DNA gets replicated, so this step is replication. And then the two chromosomes get separated, one pulled to one end, one pulled to the other end. This step is called segregation. And then the two cells finally divide. This step is called separation. Into the two daughter cells, which these are exact clones of the parental cell. So you might be wondering, when we talk about natural selection and evolution, where does natural selection fit in when these are both clones? Where does variation, genetic variation with regard to bacteria, come into play? So there is no recombination, and as a result, there's no diversity. And so does that mean there's no natural selection with regard to bacteria? Well, there is selection, and that comes through the sharing of alleles. Now, prokaryotes share alleles in one of three ways: transformation, transduction, or conjugation, and we'll look at them individually. So in transformation, an organism absorbs foreign DNA from the environment. So maybe there's a bacterial cell that gets lysed, and its genetic component, its DNA, or it has a plasmid, and that gets released into the milieu, into the environment, and an adjacent bacterium then takes it up. And that may or may not get incorporated into the new bacteria's genome, but that's transformation. So one organism absorbs genetic material from the environment. Transduction, then, one organism receives foreign DNA. I say foreign DNA, but it could be from the same species, but it's just not of the organism that we're talking about. So one bacterium receives foreign DNA via a bacteriophage, and you know what that process is all about now. You have a virus that has its own genome, and it can infect a bacterium. That's what a bacteriophage is. It's a virus that infects a bacterium, injects its DNA into the bacterial cell. That's called transduction. Conjugation, then, is when one organism receives foreign DNA from another similar organism, similar species, via a pilus or pilus. Sometimes it's called a sex pilus, sometimes it's called a sex bridge. That's conjugation. So one cell will build a little tube across, connect to the other cell, and pass its plasmid through that little tube. It's called conjugation. So here we see in this simple little illustration, transformation, the bacterium in question here absorbs the plasmid DNA from the environment. That's transformation. This may or may not get incorporated into the host genome. In transduction, the bacteriophage injects its DNA into the host. This may or may not get incorporated into the host genome. And with conjugation, one bacterium extends out this little tube, this little pilus or pilus, and the other bacterium and connects to another bacterium, similar species, and this little plasmid then is going to be passed through the tube. Now, you don't necessarily have to remember, however, either of these bacterium could extend out the pilus or the bridge. The one that extends the bridge, we call it the F+ cell. It's not male and female, but this is the F+. And the one that receives the bridge is called the F-, just to differentiate the two in this situation. Either one of these could be F1 or F+ at any time, and either one of these could be F- at any time, okay? But the idea here is that one passes alleles through this little tube into another. That's conjugation. Like a conjugal visit, right, in prison. Here is an electron micrograph of a bacterial cell. That's the sex pilus or the pilus, I prefer just, or the bridge connecting to the next bacterium. This looks very long, and actually this will retract, and this cell will get closer and closer to this cell, and then the genes will be passed through. Let's go to the video, Horizontal Gene Transfer in Bacteria by Henrik's lab, and this will explain you in a very simple way the difference between transformation, transduction, and conjugation. So another quick check, which of the following statements is true? Check all that apply. Your choices are: conjugation involves a transfer of DNA via a pilus. Transduction involves transfer of DNA via bacteriophage. Transformation involves the absorption of DNA from the environment. So let's look at them one at a time. Conjugation is when one bacterium connects to another bacterium via that little bridge and then passes genetic material through that little bridge. Transfer of DNA via a pilus, so that is true. Transduction, remember the illustration, transduction is when a bacteriophage, right, injects its DNA into a bacterium. So transduction involves a bacteriophage. So transduction involves a transfer of DNA via bacteriophage. So that is true. Transformation then is when you have DNA, a plasmid, for instance, floating out in the environment, and a bacterial cell absorbs that free-floating DNA from the environment. So transformation involves the absorption of DNA from the environment. So obviously that's true. Ta-da, they're all true. So let's talk about, real quick, prokaryotic metabolism. So prokaryotes, bacteria, are metabolically diverse, which would make sense because some live in high saline environments, some live in high sulfur environments, some live in really cold environments, etc., etc. So all prokaryotes need a source of energy. In fact, all living things, right, need a source of energy. A source of carbon. Why carbon? Because that's where your organic molecules come from. That's where your sugars come from. That's where plasma membranes come from. That's where nucleic acids come from. You need carbon in there, and a few other nutrients. So macronutrients are nutrients needed in substantial amounts. That's why we call them macro. Not that they're big, but we just need a lot of them. And you know these already. It's carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur in living organisms. You learned in 101 that even humans need these macronutrients. Macronutrients are necessary for building organic compounds, building cell walls, plasma membranes, genetic material, amino acids. Okay, you need all those things for all of these things. Versus micronutrients, which we call them trace elements. You need them to survive. We need them for life. They're essential, but we only need them in small amounts. And usually they are metallic nutrients like iron, boron, chromium, manganese. I've put the chemical symbols here. You don't necessarily need to, you know these already. You don't necessarily need to remember these. Iron you should, because that'll come back to you at some point later on in the semester. But these are all trace elements or micronutrients. Micronutrients are needed usually as reaction cofactors or carrier molecules. Like iron carries oxygen in red blood cells. Okay, let's talk about obtaining energy. So prokaryotes, bacteria, use various sources to obtain energy for ATP synthesis. And so we have phototrophs that obtain their energy from sunlight. Remember photosynthesis, you learned about that in 101. And you know that cells absorb light energy from the sun and turn that into chemical energy. And so phototrophs do the same thing. Troph pertains to energy. Photo obviously pertains to light. So this is obtaining energy from light, sunlight. Chemotrophs obtain energy obviously from chemicals, chemo, that's the chemo part. These are chemicals, natural chemicals from the environment, not obviously chemicals from the pharmacy. And production of energy that uses oxygen as a final electron acceptor is referred to as aerobic. You remember this from 101 when you studied cellular respiration, right? We called it aerobic cellular respiration because oxygen was a final electron acceptor versus anaerobic, where some other molecule is a final electron acceptor. Called that anaerobic, no oxygen. So many bacteria and archaea are anaerobic organisms. Now, many bacteria are aerobic, but many ancient, in the scheme of things, bacteria and archaea are anaerobic organisms. And it is thought that, obviously, anaerobic prokaryotes were the first organisms because the early Earth there was no oxygen. So anything that survived and reproduced had to do so without the presence of oxygen. Those had to be anaerobic organisms. All right, obtaining carbon. Now, prokaryotes use various sources to obtain carbon, obviously. And so we have this term autotrophs, which means that the organisms obtain carbon from carbon dioxide in the atmosphere. And so it's kind of like in photosynthesis, right? Plants get their carbon from carbon dioxide in the atmosphere. So we refer to plants as autotrophs. In other words, they feed themselves, right? They make glucose from the carbon that they absorb from the atmosphere via carbon dioxide. So we call them autotrophs. Heterotrophs then obtain carbon from organic compounds. Hetero means other or different. So these are organisms that obtain their carbon from different sources or from other organisms. And so here's a microscopic example of cyanobacteria. Cyanobacteria are bacteria, prokaryotes, that conduct photosynthesis. That's why you see the green here. So they obtain their energy from light. They obtain their carbon dioxide. They obtain their carbon from carbon dioxide in the atmosphere, just like plants. So here you see the microscopic specimen. And this is a big mat of cyanobacteria in some water system, some pond somewhere. And on the heterotrophic side, here you see a mixture of gram-positive and gram-negative bacteria. These are decomposers. They obtain both their energy and their carbon from organic compounds. This is just a rotting carcass of some animal, and so these decomposing bacteria come in here, break this organic material down, and absorb their carbon from there. Prokaryotes, they're everywhere. Prokaryotes, again, I said they're ubiquitous. They're everywhere all the time. But they are important players in the biosphere, both good and bad, as we'll see. So prokaryotes are important recyclers. They're important in the carbon and nitrogen cycles, as we'll see. And they're necessary for all life on Earth. If there were no bacteria, you would not be living at this very moment. Some prokaryotes are pathogenic. Pathogenic means disease-causing. Pathos refers to disease. Genic comes from genesis, which means to create. So these are organisms that create disease. Other bacteria and prokaryotes are important to human processes like food production. And so here we see the carbon cycle. I said that prokaryotes are important in the carbon and nitrogen cycles. Now we call it a cycle because it's just, you don't make carbon. Carbon just gets recycled in the environment. So the atmosphere, I call it the carbon sink. It's carbon dioxide. That's where carbon is stored to be used by the biosphere. And it's stored in the form of carbon dioxide. So carbon dioxide and the carbon dioxide gas in the atmosphere, that's the initial source of all carbon on the planet. Now, here you see terrestrial plants. Plants, of course, are going to absorb carbon dioxide in the atmosphere, and they're going to do their photosynthesis, and they're going to make their energy, and they're going to release oxygen. And then when they die and break down, right, that carbon gets re-released into the environment, and it circulates back as carbon dioxide gas into the atmosphere or into the carbon sink. Organisms like all of these animals and you and I respire. We breathe oxygen created by the plants, and we exhale carbon dioxide, which then gets returned to the carbon sink, the atmosphere. So we take in oxygen, we breathe out carbon dioxide. That carbon goes back into the carbon sink, which is the atmosphere. Human emissions like burning fossil fuels also creates carbon dioxide, which becomes part of the carbon goes into the carbon sink. It becomes part of the carbon that is available for use by other organisms. Now, carbon dioxide then gets taken up by marine organisms, like marine prokaryotes and algae, and they use the carbon dioxide and they make their energy, and that carbon dioxide gets returned. You can see the cycle here, gets returned to the land via sediment and ultimately gets returned to the atmosphere. So this is the cycle, carbon cycle, and bacteria are extreme important part of that cycle. So things to remember about the carbon cycle. Carbon cycle traces movement of carbon from inorganic sources to organic compounds and back. Inorganic sources would be the carbon dioxide gas in the atmosphere. So I call it the carbon sink. That's the atmosphere, and that's the storage place for all of the biosphere's carbon in the form of carbon dioxide gas. So that's the source of all carbon. It's a carbon sink. Now, carbon dioxide, that's CO2, is removed from the atmosphere by plants and marine prokaryotes and returned to the atmosphere via respiration of chemotrophic organisms, prokaryotes, fungi, and animals. So it's like this. The plants absorb carbon dioxide, make oxygen. Consumers, animals, come along and eat the plants, respire, breathe out carbon dioxide, which gets returned to the sink. Plants absorb carbon dioxide, and when plants die, right, they just start rotting away. They get taken care of by prokaryotes and fungi, the decomposers, which then return that carbon back to, ultimately, back to the sink, the atmosphere. Now, producers are autotrophic. Producers produce organic compounds from inorganic carbon, that is carbon dioxide. And the producers in the carbon cycle are obviously plants, terrestrial plants, and photosynthetic bacteria. Consumers then are heterotrophs. Consumers feed on, get their energy from, obtain their carbon from other organisms. So they are other eaters. They're heterotrophs. So rabbits come along and eat the grass. And then foxes come along and eat the rabbits. Or cows come along and eat the grass. And then humans come along and eat the cows. All right? That's how it works. So consumers, so the grass is the producer. The rabbit comes along and the rabbits are the initial or the primary consumers, and the fox comes along and eats the rabbit, so the fox is the secondary consumer, like that. So heterotrophs obtain carbon from producers and they release CO2 back into the atmosphere via respiration, back to the sink. And then you have decomposers finally, which are organisms that break down organic matter. So that's going to be dead and rotting plants and animals, and also waste material. So the rabbit eats the grass and the rabbit poops, and then the fox comes along and eats the rabbit, and then the fox poops. And then the fox eventually dies. So the decomposers decompose the dead grass, the dead rabbit, the dead fox, and also the poop, and return all of that material by way of carbon dioxide back to the sink, back to the atmosphere. Decomposers are namely prokaryotes, bacteria, and fungi. Another short little video from Moo Moo Math and Science, really short, really simple little a cartoon to illustrate the carbon cycle. A couple minutes long, worth your watching. All right, another quick check. What is the primary source of carbon in the carbon cycle? Our choices are plants, which release carbon dioxide during photosynthesis. By the way, that happens, right? Cyanobacteria, which release carbon dioxide during photosynthesis. That, of course, does happen. Humans, which release carbon dioxide during the burning of fossil fuels. That, of course, does happen. Animals, which release carbon dioxide during respiration. That, of course, does happen. But we're not looking for selecting all that apply, right? What is the primary source of carbon in the carbon cycle? All of these are true, but really the primary source is the sink, is the atmosphere, which is the storehouse, the reservoir for all of the carbon that the biosphere uses. Here's the nitrogen cycle. Again, it's a cycle. Nitrogen we have the nitrogen sink, again, nitrogen gas in the atmosphere that falls to the earth with precipitation, rain and snow and whatever. Falls to the earth and gets absorbed in the ground. Now there are nitrogen fixing bacteria associated with the nodules of especially legumes like peanuts that help turn that nitrogen in the soil into a form that's useful by plants. So you have nitrogen fixing bacteria taking care of plants here, okay? Now you have nitrogen fixing bacteria in the soil that then take that nitrogen absorbed from the soil and turn it into ammonia. This is NH3. You don't have to remember the chemical composition here, but this is ammonia. Now ammonia is toxic and nobody can use ammonia. So in other words, in order for that nitrogen that is bound in ammonia to get reused or returned to the sink, it has to be processed, and we'll look at that in a minute. So here's the rabbit. The rabbit eats the lettuce, poops on the ground or dies. The cabbage or lettuce here dies. So that dead matter gets decomposed by fungi and decomposing bacteria, right? And this process, it says decomposition here, but in your text, the illustration says ammonification, because all of this all this organic material is reduced back to ammonia. So this process is called ammonification, if you're curious about that in that illustration. And then you have nitrifying bacteria that take ammonia, which nobody can use, and convert it into nitrites. It says nitrates over here, and that's true. But first convert ammonia into nitrites, and then those nitrifying bacteria convert nitrites, which nobody can use, into nitrates, which is a usable form of nitrogen. Some of those nitrates, and by the way, that process is called nitrification. Some of those nitrates get absorbed by plants because they can use that form of nitrogen. That process is called assimilation. And then some of those nitrates are processed further by denitrifying bacteria, and they convert those nitrates back into nitrogen gas, which then returns to the nitrogen sink, to the atmosphere. That's the cycle. Important things to remember about the nitrogen cycle. The nitrogen cycle traces movement of nitrogen from inorganic sources, nitrogen gas in the atmosphere, to organic compounds and back again. I call it the nitrogen sink. That's the atmosphere, which is the storage source, or storage place, the reservoir for nitrogen in the form of nitrogen gas. Nitrogen gas is removed from the atmosphere by precipitation and returned to the earth that way. Nitrogen-fixing bacteria in the soil help plants absorb nitrogen. Nitrogen-fixing bacteria as well convert nitrogen absorbed from precipitation in the soil to ammonia, which is NH3, which is, again, not useful. Nobody can use ammonia, so that has to be converted. But that process is called ammonification. Decomposers are organisms that break down organic matter, like dead plants and dead animals and waste material, and return nitrogen by way of ammonia to the soil. Okay, so again, the one diagram says decomposition. Your book refers to it as ammonification. So the decomposers break down organic matter into ammonia that goes into the soil. Once again, your decomposers are prokaryotes, bacteria, and fungi. And then nitrifying bacteria convert ammonia into nitrites and then ultimately into nitrates. Ammonia is not useful, nitrites are not useful, nitrates are useful. So nitrifying bacteria convert eventually ammonia to nitrates. That's called nitrification. Assimilation is where some of those nitrates get returned to the plants, get absorbed through the plant roots to further provide nutrient to growing plants. That's called assimilation. And then denitrifying bacteria convert the rest of the nitrates into nitrogen gas, which returns to the sink. That's the nitrogen cycle. Okay, Moo Moo Math and Science has provided you another very simple, very short, little simple animation on the nitrogen cycle. Another quick check here, which the following statements about the nitrogen cycle is false. Nitrogen-fixing bacteria exist on root nodules of legumes. True. Denitrifying bacteria convert nitrates into nitrogen gas. That's true, and the nitrogen gas returns to the nitrogen sink. Ammonification is a process by which ammonium ion, or ammonia, is released from decomposing organic compounds. And we know that's true. Decomposition or ammonification, same thing. Nitrification is a process by which nitrites are converted to ammonium ion or ammonia. Ammonia is NH3. But actually nitrification, we know, is the opposite, right? That's where ammonia is converted into nitrites. So that is the statement which is false concerning the nitrogen cycle. Okay? So let's look at the idea behind our enemy, the bacteria. Some prokaryotes are pathogenic, which you all know now is disease-causing. Diseases wrought by pathogens have plagued humankind since the beginning of time. Just a quick little history lesson here, right? Some terminology. Endemic is an infection that's always present in a population. So in some parts of the world, malaria is endemic. Malaria is always there. Malaria is not endemic to the United States. An epidemic is a disease that strikes many people in a population or many individuals in a population at the same time. An epidemic would be like the annual flu epidemic in North America. A pandemic, then, is an epidemic that is widespread, usually worldwide, spreads across the world, and you are now very familiar with pandemics by way of COVID. The Spanish flu or Spanish influenza of 1918-1919 was certainly a pandemic, and millions of people died from the flu. And then the bubonic plague back in the 1300s killed, I think, a third of the population of Europe died from bubonic plague. wrought by the rat fleas, cause the plague, also called the Black Death, by the way. Now, zoonosis is a disease that affects animals, but that is transmitted ultimately to humans. And examples we know are COVID. COVID is or the coronaviruses are initially carried by bats, and when they are transmitted to humans, we call that a zoonosis. Ebola is one of those carried by bats that was transmitted to humans. And the simian influenza virus, it in and of itself is not a zoonosis per se, but chimpanzees, it's carried by chimpanzees, simians. Chimpanzees are 99.3% genetically similar to humans. So the SIV, the simian influenza virus, was easily transmitted to humans sometime back in the 50s or 60s, and was ultimately mutated into the human immunodeficiency virus. All right, so HIV sort of untechnically is a zoonosis. It's a disease that stems from a zoonosis. Not to get bogged down there. And then on the other side of the coin, we have bacteria that are friendly to us, or that we need. Nitrogen-fixing bacteria, as you have learned in the nitrogen cycle, are important to all life. We need nitrogen to make proteins, to make amino acids, which we use to make DNA and RNA. So we need nitrogen-fixing bacteria. So in that sense, they are our friends. And biotechnology is using organic systems like bacteria to make or modify products for human use or consumption. And bacteria are used often in making things like bread. Here's bread and cheese, and here is fermented meats and vegetables and wines and beers. Bacteria is used, fungi is used, of course, but we're talking about bacteria here, prokaryotes. So we use bacteria in a lot of beneficial ways, as well as, and I didn't mention it here, but I mentioned it in your objectives, your digestive system is plentiful, full of bacteria, right? 30 trillion cells, 38 trillion bacterial cells in your body right now. So, and they're not harming you. They help you out. They help you digest and absorb nutrients from your food. So in that way, bacteria help us out. Bacteria are our friends. And in bioremediation, prokaryotes, bacteria now, are used to remove pollutants like oil from oil slicks. And that's what you see in this illustration here. Preguntas, any questions, let me know. That's the end of chapter 22. See you next time.